Welcome to Evolutionary Phylogenetics number 1, in which we're just going to review how scientists classify organisms. As humans, we're faced with bewildering diversity in the living world, which can be very, very confusing. So we have a natural tendency to try and bring order into complexity, and one of the ways we do that is by giving names to things, where those names try to describe things that are similar to each other. This human process is partly fundamental to our need to be able to communicate with other humans about where good sources of food are, where good places to live are, and so on. Because if we had to use a different word for every instance of a particular source of food or somewhere to live, we would have a vocabulary that's too large to handle. So simplification is necessary, and the use of reliable labels to describe similar things is necessary for humans. Now, we've developed that process scientifically over the years, and some of the big collectors of the 19th century were well known for making large collections of organisms and classifying them into similar groups for display, such as this, either of butterflies or plants or other things. And that's a process that became increasingly scientific and less to do with gentle amateurs over the 19th century. The scientific approach was crystallized, if you like, in the 18th century by Carl Linnaeus, who developed a system that we still use today, in principle, for classifying organisms based on shared characteristics. Let's go through that process. Let's take a group of organisms, a small group in this case, that are all very different to make it nice and easy. So we've got a turtle, a lamprey, a tuna, a cheetah, a lancelet, that's an invertebrate creature, and a salamander. And we choose certain phenotypic characters with which to describe them, which might help to distinguish them. So in this case, we've chosen a vertebral column, we've chosen hair, amnion in the egg, 4 walking legs, jaws, and so on. It's really important to choose good characters. We'll come on to that in a moment. And then we score each of our type species examples for the presence or absence of those characters, and we can total up the score. Now, a character matrix like that can be clearly used by other people to reliably identify and distinguish a turtle from a tuna, for example, because of the score against these characters. We can usefully display this matrix in a tree, a relationship tree, which enables people to start at the bottom when they go out in the field, asking sequential questions about the presence or absence of characters to be able to identify the organisms that they come across in a systematic and consistent way. So, for example, the first question might be, does your organism have a vertebral column? If not, it's something like a lancelet. If it does have a vertebral column but doesn't have jaws, it's something like a lamprey or a hagfish, and so on. And using this, we can build up a relationship tree between our set of organisms that we can give to anybody that we like, who can identify these characters, and they will be able to label the organisms the same way that we do. So apart from being a useful way of helping people identify organisms consistently, does this relationship tree tell us anything deeper about the biology of these organisms? Well, I think you're so used to seeing trees, you assume it does, but that wasn't always the case. But nowadays, what we hope is that our classification trees aren't just a matter of convenience, they also reflect the phylogenetic relationships, the relationships by descent, the ancestry of the organisms in question. So, what does this mean? Let's illustrate that. In this diagram, there's a 2-dimensional plane I'm illustrating here, where the organisms in question have been laid out as black dots on that plane. And we've used the character sets of the sort just described before to group organisms into different groups. There's a pink group there and a green group. And in the 3rd dimension, going backwards in time as we go down the page, we try to illustrate the ancestry of each of those organisms back to their common ancestral points. And we can see that our groupings have been quite successful at reflecting ancestry by using phenotypic characteristics. particularly the green group there, the monophyletic group, as I've labelled it. We call it monophyletic because both of the organisms in that group, or all of the organisms in that group, share a common ancestor. And more importantly, there are no other descendants of that common ancestor that lie outside the group. So it's monophyletic. The paraphyletic group in pink is called that because although it's partly successful, all of the members of that group share a common ancestor, it's not successful in that one descendant of that common ancestor lies outside the group. And that's not a good thing. It means that the group shown in pink there doesn't fully reflect the ancestry, the phylogenetics of the organisms it's describing. One of the reasons why this happens is because there are problems with the characters you choose to do with divergent and convergent evolution. So take the convergent case first of all. You'll know that the wing is a structure adapted to flight that has evolved independently multiple times during evolution. And because it's capable of evolving multiple independent times, the use of the character wing for flight is not a good character to use in these classification tables by and large, because you'll misclassify things. Conversely, you get divergent evolution, so structures that look very different at first, for example, a bat's wing, or the paddle at the fin of a whale, or the paw of a mole, or the hand and arm of a human, all look very different, and so we could end up using them as different characteristics in the character table if we're not careful, but we know they all result from evolutionary divergence from the same pattern, the pentadactyl limb, and the pentadactyl limb is the character which is a more useful one in these classificatory systems. This problem can be best illustrated in the case of reptiles. We're generally pretty familiar with reptiles. We're comfortable with the term reptiles. I guess we think of them as cold-blooded vertebrates with scaly skins. But when we look at what we now know about the evolutionary origins of reptiles, we can see a problem. The reptiles here are the turtles, the lizards, the snakes, and the crocodiles, and they all descend from a common ancestor. That's a good thing. But the problem is that common ancestor also gave rise to birds, and we don't call birds reptiles. So that group, the pink group shown there, the reptilia, is not a taxon that has the characteristics of a monophyletic group. It's a paraphyletic group, and so doesn't accurately describe the classification of organisms in a way that relates to their ancestry and descent. So can we use something other than visible phenotypes, characteristics that can be easily measured in the field, as ways of classifying organisms that might have more robustness in terms of relating to their evolutionary history? Could we, for example, use sequence data like the amino acid sequences we're seeing in this slide, which have much more information in them, and analyze them more objectively? To see how we can do that, turn to the second module in this series.